My name's Dr. Angus Rae. I'm a life sciences microscopy specialist here at the Centre for Advanced Microscopy, or CAM. So my role here is to help out with life science researchers from all over campus at ANU and even around Australia, and help them out with their microscopy needs and their research. I work primarily with light microscopes, such as the ones in this room. So looking at the scales from about 200 nanometers up to several microns or even a few mil. We have staff at the facility who use scanning electron microscopes and transmission electron microscopes to look at things down to the angstrom level, so incredibly small scales. Today we're going to be showcasing two of our nice pieces of equipment here at CAM, our Leica Stellaris confocal microscope and our Zeiss Axio Observer compound microscope. And we're going to be having a look at a bunch of random items from around the ANU, maybe some plant samples, some animal samples, and just some random stuff from our office spaces to see what we can see what it looks like down the microscope. So the first thing that we're going to look at today is something you will have heard buzzing very loudly around campus for the last few weeks. It's a beautiful big black cicada. So here we have it. This is the eye of the cicada, and you can see each of the individual ommatidia, parts of the compound eye of the insect. One of the really special things about this microscope is it's a confocal microscope, which means we can take optical sections inside tissue. It has hardware that removes out of focus blur. So we can image from the top of the eye deep within the eye. And we can even take a set of images and reconstruct it in 3D. So the next thing we're going to look at is some printed paper from the ANU reporter. So we've cut a small square that's colorful from one of the pages, and we're going to have a look at it on our Zeiss Axio Observer microscope. Okay, here we have it. This is a printed color on a page, and we can see each individual dot of cyan, yellow, or magenta that the printer lays down to make all of the visible colors that we can see in print. This is at 50 times magnification. Okay, our next specimen is a flower. Common weed found across campus, and what we're going to do is see if we can get some pollen out of the flower. Here you go, individual grains of pollen on the microscope. This time we're looking at around or more than 400 times magnification, and the different colors here are what's called fluorescence lifetimes, and you can Google that if you're interested. Our next specimen is a feather, single white feather, probably from a cockatoo or a corella, and we're going to look at it with 100 times magnification. All right, here we have the feather at 100 times magnification, and what we're looking at is naturally occurring autofluorescence again, and you can see the individual interlocking barbs of the feather shafts. Okay, our next specimen is a piece of cork. Now, cork has a special history in microscopy because the word cell was first used to describe what a microscopist saw when looking at a piece of cork. So here we have it, cork under the microscope. So what you're seeing are individual plant cells in what looks like a honeycomb pattern. In this case, the cells are dead and we're just looking at the cell wall. And the contrast that we're seeing comes from naturally occurring fluorescence. I love it. You get to see the hidden detail in stuff from everyday life. And we get to work with a huge range of different researchers. So from plant cell biology to cancer biology to virology. So getting to see the tiny details of these biological structures down a microscope and getting to assist with all sorts of different kinds of research and important research is really exciting. I love doing it.